I would like to call upon stage Professor Deepak Kumar from DBG IISc and convener of Longevity India Initiative to have the first keynote address. Over to you, sir.
Thank you, everyone. I'm back again. And I was told that I'm supposed to be here the buffer between the actual talks. No, not actually. I will give a proper scientific talk also. First of all, thank you, everyone, for taking time, 3 days of deliberations to come to this meeting. And I must also say that this is the first time we're doing this type of meeting where we have academia, clinicians, industry, and policymakers under one umbrella. So we're all looking forward to discussions during the next 3 days. We are quite excited. And as a part of the organizing team, I take complete onus of anything which goes wrong in the next 3 days. But if everything goes right, there are a lot of people on the ground who have done fantastic work to make this meeting happen. And if there's anybody who faces any issues, please feel free to ping me, and I'll see what can be done. So now, coming to the technical talk, which I'm supposed to tell you about what my group has been doing. So my group is called Saini's Lab, and it's an acronym, which is signaling in aging, infection, and inflammation. And this acronym was coined by one of my students, and that idea was to capture what my lab does. I'll take you through the journey. They're not a lot of technical storytelling I'll be doing. I'll be just giving a glimpse of what my lab has been doing from last 15 years at Indian Institute of Science, and maybe before that also, to give you an idea how it actually led to genesis of Longevity India, how and why we landed with Longevity India in the first place. This is my journey. I started as a microbiologist. I started looking at bacterial communication. And I think once I started, I got hooked to communication. I wanted to see how cells communicate and make decisions in anything which they do, whether it is pathogenicity, virulence, adaptation, survival. We were basically looking for how communication works. And after I finished my PhD, which was in bacterial communication, I moved to a slightly higher order of communication to look at GPCR signaling, which is the receptors in our body, which talks to the environment and takes decision for almost all physiological processes in our body, right from seeing light, taste, pain, neuronal connections, serotonin, dopamine, everything is linked to GPCR, and GPCR is also the highly druggable candidate. People like to use GPCRs to drug and cure various disorders. And that was what I did for a few years before I joined IISc. And when I joined IISc, the question which we started asking was, or the first question which we asked was, well, because I have a history of looking at communication and looking for GPCR, I wanted to look for a receptor, a GPCR molecule, which we can use to target aged cell. It was a very simplistic viewpoint. Is there a receptor in aged cell which we can use to target cells and change the way this aged cell behaves? And that's where we looked for a particular receptor. A very interesting receptor came to play, which we call it CXCR4. I'll talk about that a little later. But this started the aging research in my group. We were looking at very fundamental processes, completely cellular, looking at molecules, pathways, which we love to do, because that's what we are trained to do for a longest time. So we look at molecules and cells and systems, which led us to look for molecules or genes which regulate aging. And we started looking at entire human genome genes, and we started removing them one at a time to understand how we can control aging. And that led us to mitochondria, which led us to looking at nitric oxide, and we started looking at the role of aging on infection. Then, as the journey progressed, we got interested into collaborative research. A lot of people had fantastic tools in the Institute of Science. We started developing 3D model systems in aging, and more recently, we've been interested in how cholesterol, a key molecule, which is called the bad molecule for a lot of healthcare practitioners, is actually, we found that a very interesting relationship of cholesterol, and our studies suggested it's probably not a bad molecule, it's a good molecule, and the oxidized cholesterol is the bad culprit. And that journey more recently led into emergence of Longevity India. So coming out from the beginning of this story, when we study aging, the classical model system of aging says, you take a cell, damage its DNA. And the DNA's damage is going to undergo aging, or we call it cellular senescence. And people love doing that by various different approaches. We like to talk about shorten of telomerase, oxidative stress, genotoxic stress, activate a cancer oncogene, and we can trigger senescence. And this accumulation of senescence in the body, which has been shown to be responsible for aging or systemic aging. Now, we were asking a very simple question. We started the question by asking, said, all these ways by which we can trigger aging, are they actually interlinked to a simple process, which is the DNA damage? Now, can I delink DNA damage from reactive oxygen species? That was the first question, because reactive oxygen species considered the main culprit behind aging. It's a reactive oxygen species which damages DNA. What we did was we actually took an analog of a molecule which is found in DNA, a thymidine analog, which is bromodeoxyuridine. For the people who are non-scientific like us, who'd like to look into details of molecule, this molecule goes into DNA, sits into the DNA, but DNA machinery which replicates DNA does not like this molecule because it's not the true sense of molecule which makes the DNA. It's an invader. So it triggers a process which we call as DNA damage response. It asks the cells, I don't think this is the right DNA. We need to stop replication and trigger damage response. What happens is that we can quantify this damage response. This is damaged cells, and you can see all these interesting lesions or foci, and you can count them and quantitate them. And this is the normal cell. As you can see, this cell which is treated with this agent shows numerous small puncture foci or damaged lesions. And these damaged cells actually enter aged phenotype. Now, at a cellular perspective, a damaged aged cell has a very interesting fate. It sits in your lab without dying, without dividing. So it's like the old people who sit under the tree in the ancient system who actually just sit there for years altogether. And sometimes you laughingly say, I don't see these guys doing anything. They just sit there and pass comments to everybody who passes by. And that's what exactly the aged cell is. It just sits there, does not divide, does not die, and just keep generating this inflammatory milieu or inflammatory cytokine, which starts triggering systemic aging. And when we use this system, we could actually find that system mimics almost all the features of aging cells, which includes arrest of cell death, changing in the extracellular milieu, inflammatory cytokine, and loss of cell proliferation. And using this model, we ended up proving something very interesting, that reactive oxygen species does not necessarily have to trigger aging. It plays a role in maintenance of senescence stage, keeping cells alive without killing them, but the cells can become aged if the DNA is damaged independent of reactive oxygen species also. However, in our system and metabolic problems which we face, reactive oxygen species happens to be the main molecule which damages our DNA and causes enhancement of aging. So while looking at this aging cell, we had an interesting model system. We were asking a question, is this model system help me reach my receptor which I want to target? So we did find one receptor. When we did a large-scale analysis of genes which are changed or proteins which are changed, we found one receptor which is increased in aged cell. And this receptor, which we call as CXCR4, it's a cytokine or a chemokine receptor. Interesting molecule, expressed in every known cell in our body. And what we found, this receptor is present at a higher copy number in almost all aging model systems which we use. Some of them are our favorite lab models, some of them are primary cells, young and old. In every system, what we found, this receptor was upregulated. It means it was present in higher copy number. And this receptor, what we ended up proving through a long drawn experiment, which took 5 years of a PhD student's career, to prove that this receptor increases inflammatory responses in the cells. And this is the model which we came up with, that in our body, we have all the cells which are young. Some cells undergo damage and become old, and these are the old cells. And the moment these cells become old, the receptor, which is this CXCR4 receptor, is present in high copy number in this cell, which increases their sensitivity to the ligand because the copy number is more, and it starts generating inflammatory cytokine locally around this damaged cell, so immune cells can home and clear this damaged cell. Now that's what happens in our body all the time when we are young. Damaged cells are generated, our immune system comes and clears it. But what happens when there are too many damaged cells? That's what happens when we start getting old, because there are too many points of clearance machinery is needed. It's like a super dirty place and few cleaning machinery. This leads to accumulation of damaged cells in our body, which leads to systemic aging. So we found this receptor to be very interesting in terms of clearing through by the process of inducing inflammation. Now, while we were making this model, we had fantastic data. We saw this receptor is on, inflammation is on, you recruit immune cell. But by mistake, one of my students actually forgot to keep the cells back into the freezer, and she left the cells in an incubator for many days. And what we found that after some days, this receptor does not respond to inflammation. It actually stopped triggering inflammatory responses. It actually changes behavior completely. Now, we were very intrigued. We were intrigued for a very long time. For such a long time, we actually put this experiment also in the freezer. We said, okay, this is something very bizarre happening. We really do not know why a receptor suddenly changed from an inflammatory inducing receptor to an inflammation inhibiting receptor, which is in the late stage of senescence. Now, this raised very, very interesting problems for us, and we kept sleeping on it for a very long time till next student in the lab joined, Suramya, who actually said, let's look at if something is wrong in this pathway. Now, I am sure all of you have interacted with Suramya. She's been shooting all kind of mails to everybody out here. Now, that's what she does not do in real life. In real life, she's actually a scientist on the bench trying to do some sense of this receptor. So she looked at that if the receptor is intact, it was yes. If the receptor was triggering signaling changes, it was fine. And then we asked the question, what exactly is wrong with this receptor? This is a movie. I hope this movie plays with this. It does not play. So this is how it's playing. So you can see these Christmas lights. They look like Christmas lights or Diwali fireworks. So this is the normal cell, that when we activate this receptor, these fireworks or these calcium oscillations come inside the cell. And they keep coming because that's what receptors do. They trigger calcium release and they reabsorb calcium. And in a healthy cell, these fireworks keep happening. But in the aged cell, we do not see these fireworks happening. And if you plot these fireworks, you can see these profiles, and these are the profile of the aged cell. And we found that the receptor was not able to trigger the signaling as it was supposed to. And after a lot of thought process, we said, this receptor loves cholesterol. It wants cholesterol to stay on the membrane. It wants cholesterol to stay active. Let's look at the cholesterol molecule. And our assumption was very simplistic, that if the aged cell has high oxidative stress, the cholesterol is going to undergo oxidation and form this notorious molecule called oxysterol. Now, oxysterol, everybody knows that it's actually a bad molecule. But we wanted to prove that this is the bad molecule for aging also. And what we did with this hypothesis that increased reactive oxygen species converts our favorite molecule cholesterol to oxysterol, which is the bad cholesterol, we actually ended up quantifying in the aged cell. And what we found in the aged cell, cholesterol is low and oxidized cholesterol is high. And these are all different types of oxidized cholesterol. Cholesterol is a funny molecule. There's a tail, there is a head, and oxidation can occur at multiple places. And what we found that almost all the places, oxidized cholesterol was going high. But that doesn't still prove anything. It shows the cholesterol is oxidized, which is expected. It still does not link to our CXCR story. What we did, we took young cells and we threw oxidized cholesterol into these cells. We did not wait for them to get old. We just threw it and we asked, does that affect my calcium signaling? And the answer is yes. Oxidized cholesterol actually impairs or knocks out calcium signaling. So now we have a very interesting hypothesis. Just the presence of oxysterol is sufficient to change the way receptors on the membrane of a cell works. And this is a big hypothesis picture. This is normal cholesterol, oxidized cholesterol. This is what a normal receptor in a cell doing, enhancing inflammation. In the aged cell, it is anti-inflammatory. Now, if you think this carefully, this observation tells us something very fascinating. GPCRs are drugged. Basically, all the known drugs, most of the drugs, right from histamine receptor antagonist to dopamine to serotonin, we actually use them as drug targets. And if you think from this observation, we find that many of the drugs do not work that effectively in aged people. What if this is the culprit? What if we have been looking at druggability of receptors in the young population? Because that's what we do. When we look for drugs, screen for drugs, we do trials in young people. We actually exclude old people from trial because they give very heterogeneous data. This is a reason for heterogeneity. Is this the way we can actually address the requirement of drugs and druggability in aged population? What we are saying that we really need to relook at receptors which need to be drugged in aged population, because if this kind of switch is happening, we are actually either under-drugging them or over-drugging them, or we probably triggering some pathologies which need not be there in the first place. So this is what we have been doing with GPCR. I'll quickly shift gears now. This is some screen which we did. We knocked down a whole bunch of genes in our cell system to see which gene, when you remove, can switch on aging without actually any damage from outside. And one of the genes which we found was here, it's called TIM50. It is a phosphatase means which removes phosphate from proteins, but this is actually a mitochondrial protein. And what we found that in normal aging, this protein expression comes down. And a lot of studies, again, Amrita was doing this study, and a lot of studies actually reveal that when you actually stabilize this protein, which is actually one protein which links our favorite anti-aging molecule sirtuin to TIM50, we showed that sirtuin actually regulates the expression of TIM50, and when TIM50 is stabilized, the cells actually do not enter senescence stage. And we also found a small molecule stabilizer for this TIM50. Now, if you give this small molecule stabilizer and you insult the cell by reactive oxygen species or any other agent for aging, they do not enter aging, or they slow down significantly. So this is one of our attempts to use pharmacological inhibitors to address aging and age-associated pathologies. Now, this is where we ventured into trying to understand that if this is indeed true that mitochondria is the key driver for aging, what if I damage just the mitochondria? I leave my nucleus out of the picture. So one of my students, Abraham, this is still unpublished, he actually developed a model by damaging the mitochondria selectively by very low dose radiation treatment. And he showed that if he can stop mitochondrial injury, prevent mitochondrial ROS generation, the nuclear DNA does not get damaged, and you can prevent senescence from onset. So this comes full circle to a lot of popular theory of aging, which says mitochondria is the key culprit in aging. It's the mitochondria which dysfunction triggers aging. And we have very interesting evidence to show that indeed mitochondria could be the major driver of aging. Now this leads me to a lot of collaborative studies which is going on with various faculty in Indian Institute of Science. We've been looking at, with Siddharth's group, that if in the aged animal we trigger an infectious agent, here we use salmonella and tuberculosis, do they actually succumb as the classical thought process says? What we found was reverse. We actually found the aged animal or aged cell system which we're using were resistant. The bacterial burden was lower in the aging. And that was primarily because they had high nitric oxide, which is a key antibacterial, antimicrobial agent. And we block this nitric oxide, actually the infection goes up. Interestingly, we found that even though the burden is low, the actual effect, basically mortality, was not lower because the aged animal or people die because of unresolved inflammation rather than the bacterial burden which is in their body. We also have developed cellular systems, which is in 3D culture systems, and developed biomaterial with Professor Kaushik Chatterjee's group, which we use to study aging, and we found the aging which we use in lab in 2D can be an exaggerated response, and what we get aging in 3D is actually a suppressed inflammatory response, similar to what we see in our body. We also developed some biomaterial for aging in which we have introduced agents like ceria, which quench reactive oxygen species, and that's why making this material more useful for aging people. This is where we are right now. We're looking at a lot of interesting aspects, but again, fundamental, looking at DNA damage, proteostasis, mitochondrial dysfunction, biomolecule oxidation, and biomechanical and drug screening. While this was happening, a lot of collaboration was happening, we realized probably looking at cellular systems, just cells and model systems, animal, are not sufficient. We must take the leap of faith and transition to look at human studies. And that's where we actually thought about transitioning to a multidisciplinary investigation and move on to human studies. And that led to a formation of Longevity India. This is the global landscape, a lot of interesting work done globally. We have David here from Buck Institute. We have other speakers will be here representing the global community of longevity research, and we established Longevity India in IISc. So I'll take you a quick journey about what Longevity India, how it came into being, and what was the genesis. It was around in August 23. A lot of us, people from various disciplines, including biological sciences, engineers, data guys, come together, and we start thinking about can we use our strength of various different disciplines which we are trained to actually look at problem of aging, because it needs, as a lot of speakers said, a multidisciplinary, multi-viewpoint. And using that, we actually projected this idea to Mr. Prashant Prakash, who was generous enough to give us the seed funding, and which led to actually nucleation of this project as Longevity India. In April 2024, we launched Longevity India Initiative, and 2024, August, we had the first industry conclave. A lot of people here in the audience were part of the deliberations. It was an interesting presentation where we did not give too many academicians a platform to talk about, even though it was an IISc. We had a lot of industry because we wanted to hear how industry views longevity. How do they see things are done in the industry setting, because there are a lot of true and not so true things which are happening in industry in addressing longevity or aging research. And in 2024, we launched Bharat Study. Bharat Study is an acronym. I'll come to that a little later. This study aims to look at healthy aging in India by multi-parametric viewpoint. We have got multiple studies going on in India where people have been looking at gene relationship with aging. We have this study where we talk about Indian Genome Atlas. We have also one study which has been done by Avasthaj and Avastha Genome, which actually talked about looking at genomic of Indian and Parsi population to see we can find some correlates. But what we are trying to do is collect a lot of multi-parametric data. And then we have recently got ICMR CAR, and here we are at RISE. Now, this is what Longevity India aims to do. We are actually trying to bridge science, medicine, technology, and society to develop a hub in which everybody can be linked and try to address next generation problems of longevity. This is a team. I'm not going to read the names. They're all in the audience. Find them here and talk to them. But what's the strength of the team? They're all specialists in one part, one organ of the human body, one data science, one microbiome expert, biomaterial guys. What we did was to put everybody together so that everybody can lend their strengths to address one common problem, which is aging and longevity. And please feel free to talk to our colleagues who are in the audience with you. And this is what Bharat Study is all about. Bharat Study is an acronym, as I mentioned. It is biomarkers for healthy aging, resilience, adversity, and transition. And if you look at this acronym, it pretty much summarizes what changes happens in our body during our lifetime. In this study, we are sampling healthy Indian population, rural, urban. We reach out to community level to get healthy data, and we collect a lot of data, almost 2 million data points, which includes functional changes, cognitive, metabolic changes from serum proteome, serum metabolome, serum metal ion, epigenetic, gut biome, biophysical changes in the blood cells, immune profile. A lot more data is collected so we can make a footprint of a healthy Indian so that we can understand what are the trajectories of aging. What Longevity India wants to do, this I will not go through this slide in detail, but our idea is in the long run we can imagine, investigate, innovate, and implement various aspects of healthy aging by using stakeholders which are scientists, industry people, clinicians, and investors. So we can develop a completely new ecosystem where we talk about aging with various lenses. And this is our long-term vision. This is the longevity vision. What we have shown here in this side is what we have strengths in. We want to refine India's specific standards of health, Bharat standard, nutritional, screening technology, drug development, novel therapeutic targets, biomarker discovery, and cellular model systems. And this is our vision, that this is where it will have societal impact, reduce hospitalization, dignity in aging, preventive health, improve quality of life, and societal engagement. For a country like this country, India, which is so huge, a crowded hospital is not a good sign for the country. To have a good understanding of a country's healthcare, we would like to have empty hospitals, where people only go when there is actually a need. We talk about sick care. We should not go to hospital for health care. Healthcare is something which we should understand better so that we can avoid people from reaching hospitals when they don't need to do. And I'll stop here, and this initiative is supported by Prashant as a stage, and right now we've got ICMR support to continue this initiative. My lab has been funded by ICMR, DBT, DST. Longevity India has also received fellowship support from Ms. Kiran Mazumdar-Shaw, donations and CSR support. These are lots of collaborators, and collaborators are a very integral part of modern science. We can choose to do everything in our labs, but we really need collaborators from industry to make this vision happen. A lot of these companies are giving support to us by giving us access to technologies on priority basis, access to technology at low cost, access to platforms which are still not public, so that we can actually develop this longevity data set in a very meaningful way. And we are looking for support. The support is very critical for an initiative like this. No matter how much funding support we get, for each individual, if we spend about 2 lakh rupees to collect data, we really need a lot of support to keep it going. And to prevent it from derailing, we are looking for support in various flavors. We're looking for people who help us data collection, fellowships, volunteers. Every healthy person is a potential volunteer for us for collecting health data, collaborative research projects, internship, donation, and we have a very active society and community outreach channel. And I'll stop here, and I'll be open to questions. Thank you very much. Yes.
Excellent presentation. While you're collecting data in the Bharat study at different dimensions, is there any way you are doing phenotypic stratification as well to, look at the different groups and how they fare on those parameters?
Yes, we're collecting huge amount of metadata. So a lot of phenotypic data, which includes their diet, lifestyle, their prior history, their family history, their habits and everything. So there is almost 75 pages of metadata which we're going to be collecting as part of the study.
I
So
would
it means
let's-
it allows us to stratify.
Yeah, I think one of the ways by which Ayurveda can contribute is phenotypic stratification of healthy individuals by Prakriti, and whether you see, and there are many other parameters also by which you can clinically stratify individuals to look for underlying molecular differences.
We'd love to talk to you about that. As I said, it's a hub model where we love to have people suggest how we can improve our data collection.
Deepak, very intriguing finding that cholesterol get oxidized during aging. So have you looked at how it's affecting the overall rigidity of the membrane and how it's affecting the raft formation and then overall general signaling transduction which might happen from membrane to the nucleus, how that's dynamics is changing?
Fantastic question, Rupesh. I will not answer it because you need to listen to Suramia's talk. She's going to give all the details about what changes in a cell when you get exposed to oxidized. She's got some humongous amount of data, and I will not steal her thunder, so let her talk to you, and you can...
Well, my question was also extension of what Rupesh has asked. First of all, excellent presentation. There are situations where excessive inflammatory response is a problem.
Yes.
And oxidized cholesterol, are they already used as some kind of
Yes.
dampening
Yes,
agent?
actually, oxidized cholesterol seems to go up in a lot of cases, like from neurodegenerative disorders, including cardiovascular disorders. We do see oxysterol enhancement to be there. The problem is there's so many different types of oxysterol. And to figure out which one goes up in which system, that requires some high resolution mass spectrometry, and that is something which we're in process of developing. So yes, oxysterol is a known culprit. We still do not know why it comes into the system.
Sir, actually, the epigenetic model, especially a reaction of the environment to the species with respect to the mitochondrial changes, that Ayurvedic applications, a reaction of the environment to the individuals, both in health and also pathology manifestation, does it have any impact? Because Dr. Tapas is working on the same, some of the things at JNCS, we are associating with using the Ayurvedic, some of the principles rather. Does it have any impact? What is your comment upon?
So this is something which I think Dr. David Furman is an expert on. He'll talking about that.
Yeah.
Exposome is everything.
Yeah.
You have to account for what the environment is trying to tell you. And what exposome does to your body is what aging is going to do in the long run. So yes, environment plays the important role in your aging trajectory.
Because Ayurveda may throw some more light.
Yes,
upon.
absolutely. Ayurveda will throw very interesting insights into this process.
Hi, Professor Saini. Here, I'm here. I am Ritu Tamap. I'm from Unilever. So, excellent work on the senescent cells. And we all know that how ROS, radioactive oxygen species, are damaging the normal cells, healthy cells, and like after aging, it is getting senescent. And there are many ways to treat the senescent cells, for example, senolytics and xenomorphics. But is there any way to target the senescent cells to block the receptors, like which has the increased copy numbers, or to get the calcium signaling back into the picture? So your comments.
This part is something which we have already published. We have got molecules which can reduce the expression level of CXCR4 receptor. But see, if you do that, the question is always in the senescence community is when and how much. When do you do this? When do you stop it? Because if you're young, this is the healthy response. You want senescent cell to be removed because immune cells will come and clear it up. So do you want it to stop it before the immune system actually clears it? And how much should you suppress it? So that conundrum will always stay, that do you want the aging responses to be stopped too early? We have not actually answered that question yet.
But because there will be no inflammation, as you have already shown, how the immune response will happen?
So the more inflammation is very local. The damaged cell generates a very local gradient of inflammation around it so immune cells can home to it. So it's very local. Once it's unresolved and there are too many damage points, then it becomes systemic. So so the question will always stay is when do we know when to give that drug or entity to prevent inflammation from happening?
Thank you.
Thank you.
One more question only. We have 30 seconds left.
Deepak, very nice work and presentation. See, when you induce a renal ischemia, like an acute stress, one of the things that happens is that cells accumulate cholesterol. So there is about 10 to 25-fold increase in cellular cholesterol content after an acute kind of an insult. So now, after that happens, when you go back and redo the insult to the same organ, the cellular cholesterol accumulation in the cells actually protects the organ from a second wave of injury or an insult. So this has been shown that if we deplete the cholesterol, then this resistance mechanism after an acute insult is prevented. So meaning that, cellular cholesterol accumulation is actually a protective response.
Yes.
Following an acute insult, for further kind of a thing. So it's very interesting in the context of the senescence models that you see, the accumulation of cholesterol, may be an acute response to stress to protect the cell. But then it becomes maladaptive, right? That's what it looks like. Yeah.
So I think the problem is when we measure cholesterol, we do not have means to distinguish between cholesterol and oxysterol as of now. So that debate will stand unresolved till we figure out that way. And once we do that, I think we'll get a lot of beautiful insights into cholesterol role. There's a very old paper in 1940s which says cholesterol is an antioxidant. So maybe perhaps that was the right way to look at it. So I'll stop here for the lack of time, and thank you very much.